Hi guys, yeah, we're all hearing. So this is the last talk of the day, so I'll try and not keep you too long, but what I want to try and do is bring a different flavour to the day and say what we're trying to do, we're trying to create a new article type and a new platform for the publication of antibodies. So F1000Research is, we're a publisher, but we're very different from all other publishers and other journals, and I would say we're quite on the left compared to them. And we kind of go on these 4 pillars that keep us going, which is, the first one is speed, so you can publish an article in 7 days, so we do post-publication peer review. So I think our record is 37 hours from submission to publication. We've had an article. And as I say, we do post-publication peer review, and it's completely transparent and open. So everyone knows who's reviewed the paper. We kind of encourage discourse between the scientist and the reviewer. And we found that it's brought a bit of civility to the process. The reviews have been a bit more constructive, and it's probably the way peer review should have been, and not this kind of closed, hidden process that we get today. We're also got no barriers, so there's no editorial bias. So there's no gatekeepers, there's no peer reviewers who say you can and cannot publish. It's published straight away, and then the work is judged on its merit afterwards, and again, that's all done transparently. And another key thing that we're doing, and journals are moving more and more to it, is open data. So I was going to use the preclinical cancer biology studies like David did earlier, but reproducibility is a big problem, and it's probably because the data isn't available within a paper. So you've just got to take into consideration the STAP cell papers that were in Nature this year. For some reason they managed to get published, that you could put a somatic cell in some acid and boom, you've got a stem cell. Everyone in the community thought that's not possible. But it managed to get published, and obviously we actually published a refutation of it, and which had all the data, and it clearly proved that it was wrong. So even higher tier journals are kind of providing this information that may be false. So here we say that you have to give us the data. There are obviously some sensitivity issues surrounding some, but that's what we ask for. So what should publishing be, and what the kind of ethos of our company is, of our publisher is basically, I think next year is going to be the 350th year anniversary of the first publication journal. So the Royal Society's Philosophical Transactions published one of Sir Isaac Newton's papers, and what we had to do was fit the science into the article because there was a printing press. That's how we had to do it. But is that how we should do it nowadays? So let's just think of something else that was about in the 17th century. You've got to think of a newspaper that was also built so the news could fit the printing press. But think of the news these days. You've kind of got real-time news, you've got collaborations with readers talking with news people, multimedia. It's really moved on. It's embraced the digital age, and what we think is, why are we still doing science fit the article when the article should be fitting the science? The way we publish should be on a platform that really makes the science stand out. So that's what we're trying to achieve. So let's just go through a quick traditional publishing. So you'd submit your article, and then you'll enter the virtuous circle of peer review, which can go on for 6 to 12 months, and I know some people refer to it as a gauntlet or a battlefield. It's really what goes on there, and is said can be hidden, and then your publication can come out. But I mean, you can sometimes be missing the boat, and your research is taking too long to come out. Our process is, you submit the article, it goes up straight away. The data is put in a repository, so if it's a gene, it will go into GeneBank. If it's a protein, it will go into the protein database. If it's more generic data, we put it in something like Figshare or Dryad. We just make sure that data is always going to be publicly available. And then once this is published, then the peer review process happens, and obviously there's still going to be comments where you're going to have to make a revision, a figure might not be right, you might have to have a paragraph, but that's fine, that can all happen in the open, you can see why you've done that, and you just create a new version which sits on top of the original version. So then you've got a whole catalogue of what's gone on with that work. So a little demonstration is this paper here from the University of Edinburgh. The author wanted to get out very quickly. He had other competing labs going on in America who were doing the same kind of thing. He published it on the 25th of December, and the first reviewer instantly said, I think you're missing something. So he did a new version, which counteracted what he said. And so this reviewer's major reservations turned into okay, I approve this now. We had another reviewer who instantly approved it, probably because he'd seen this one as well. But then another reviewer came in and had something else that he wanted changing. And then the third version was done, and so altogether we have now got a referee status of 3 ticks. And if you have 2 ticks, or 2 ticks and a question mark, you'll be indexed in PubMed. So none of these studies will be indexed and available in bibliographic databases until it's met the criteria of the reviewers. So an antibody validation article type. So a lot of been talked about today about what's going on, but antibodies are the most common reagent in a lab. I think I read something in The Scientist that 88% of labs use laboratories in biology and medicine. And there is this common perception that there's a lack of standard guidelines when it comes to manufacturing and validating. So in an ideal world, we kind of think that researchers would like a confidence in a particular antibody. As everyone has said today, specificity, selectivity, and reproducibility is key. And then it would be useful to have some information based on actual use cases from the scientific community, people who are working on things that you're working on, validating theirs. So what we've done to tackle this problem is we spoke with a few people. So Andrew Chalmers and Matt Helsby from SciTab and May from St John's Laboratory. We all sat down and we thought, what's the scope? Can we create an article? And these are the main things that we covered during that. Was what's the viability? What would be the costs, the incentives? Who would be interested in this? Who would be potentially conflicting in this? We then had to, most of the talk was around what would we need to put in the paper, so the different types of validations you can have. We wanted to create a set of flexible guidelines, which I think I've put on all your seats. Please do read at your leisure, but we wanted to keep it flexible so we're getting the information that we need, but we're not being too burdensome, or obviously David's rigorous. I don't think we can put them in straight away, but what we want to do is try and get a nice standard way of validating. And then we thought, by doing this, we also need to disseminate it properly. So how are we going to make it discoverable? How are we going to make people use this? So that happened in May. We came to September now, and this is what we've done so far. We've got a set of author guidelines, like I said. We've created a diverse editorial board and tried to have some industry and some academia in there. And we've also created a dedicated antibody validation collection which went live today. And the first articles, we have an opinion piece from Jan about commercial antibodies, and we have a couple of validations, one from Simon and one from Will. So people are already starting using it. So this is what the collection looks like, and there's some articles in there. There's a description of what we're doing. The editorial board and everyone is mentioned, and this is the URL to it, and we're hoping to get lots of validation studies published here. So like I said, we have an editorial board. The guys that we spoke with first, they're our first sounding board if we have ideas, and they obviously are well placed to tell us what we can and can't do. But then we also use this international advisory board to kind of bounce ideas off and hopefully help us choose the right people to review papers and whatnot. This list is obviously not exhaustive. We want to include everyone and anyone, so if you want to be a part of the board or be a part of this collection, then please do let us know. We want this to be a community thing. And the article guidelines too, we've sent it round maybe 10, 15 people who are involved in antibody validation. However, there are going to be other ideas, maybe more from industry and more from academia. We want to hear all the ideas. We want to try and make this as inclusive as possible. So whatever you read, if you do disagree or agree, please let us know, and we will happily accommodate you. So the next steps, so this is kind of going to lead into the Q&A as well, really. We've created this space, and F1000Research is a very agile company. We have developers, a huge development team who look after everything, and we do things monthly. We can build things, we can take things out, we can add. So what we want to do is create a community feel where everybody can get involved in this. So I suppose the questions I would like to ask, and if you think that, this is a good idea, how would we go about promoting this new article type and collection to the wider community? I mean, obviously this is a perfect place. You guys are all into the antibody validation and think it's an important thing, but the common scientist in the lab might not understand where to find this. I don't think we need a stick, but I do think we need carrots. I think we need to instill the idea that publishing a validation is worthwhile. I think, did Simon say he did 250 validations for his last paper? So, I mean, PhD students do this, and to have a tangible, citable publication from that, I think, is an incentive. But is there anything else? Do we need to give out iPads? Do we need to give out bikes? And also, I just want to look at a way we can help grow the content of the collection. So I think CiteHab said there's 2 million antibodies, so there's potentially 2 million validations that can happen, and it would be great if we can have a place that has all the data, all the methodology, which has all the manufacturer details, proper Western blots, every single one of them, not just cut, fudged images, all the data that we need, and somewhere where people can say, I have validated this, you can go and look at it here. So yeah, there's a bit more. So please feel free to give us feedback on what we've done to date. Contribution, we're happy to have any input. Are there any potential pitfalls that you might see in this? I mean, does it work for industry? Does it work for academia? I can see the benefits for both. And again, I think this is my biggest question, is how do we best reach the community with this if we did want to really push an antibody validation? So with that, let's start Q&A.